I did bring exams today to hand out, and so I'll hand those out at the end of the period. I know I've learned my lesson in previous years that you do that at the end of the period. You at least have some semblance of people's attention, and I'll say a word about the exams when I get to that point. I have posted a key outside my office, so if you want to come over and see the answers, it is posted at my office. Okay, so turn our attention to coming back to techniques. So we spent some time previously talking about techniques relating to proteins and large molecules, and we've learned some other things now relevant to nucleic acids and so forth. And so it's appropriate that we spend a little bit of time talking about some techniques related to molecular biology, which is what this is about. So as we look at biotechnology techniques, one of the things we talked about previously had to do with separation of nucleic acids. And separation of nucleic acids most commonly occurs on agarose gels, although for very small fragments of nucleic acid. And by small, we're talking typically about 300 base pairs or smaller. For smaller ones, we use polyacrylamide gels, but we don't use SDS because we don't need to for nucleic acids. Separation by gel electrophoresis, of course, involves the fact that the backbone of the nucleic acid is a polyanionic substance, meaning it's phosphates, and one phosphate for every nucleotide in the backbone of that. So all those phosphates are negatively charged at physiological pH, and so when I put this into a chamber such as I loaded in this well, the negative charges are repelled by the negative electrode up here. So they move as fast as they can away from that. And since the ratio of the size to the charge is constant, because there's one more phosphate for every nucleotide that's there, these guys separate then solely on the basis of size. So the smallest ones at the bottom, largest ones at the top. And we talked about that previously. Now, one of the things I didn't point out last time, I will say here, is that when we run a gel, we don't typically see this, okay? Unless we do a couple of things. So to actually visualize fragments of DNA in a gel, we have to stain them. And so commonly there are a variety of stains that are used. In the past, one of the more common ones was a dye called ethidium bromide. This is a dye that actually gets physically in between the base pairs of the DNA. And it has the interesting property that when it is in that configuration and UV light is shined upon it, that it fluoresces very brightly. So you can actually see the fragments when you do both of those treatments that I just described. Now, one of the things you might wonder how it is that we get specific fragments of DNA. What do we do to get those? Because we know that chromosomes, for example, are very long. We know that plasmids, which I'll talk about in a bit, are fairly small. And some of those fragments that you see on there are even smaller than that. So how do we generate fragments of DNA? Because it's typically DNA that we're separating on a gel. The way that happens is by use of enzymes called restriction endonucleases that cut DNA at very specific nucleotide sequences. Okay? So restriction endonucleases cut DNA at specific sequences. And they will cut only at those sequences. So if I have a piece of DNA, for example, that has the sequence GAATTC, then the restriction enzyme known as EcoR1 will cut at that site, and it will cut on both strands. And it cuts at the GAATTC. And you might think, well, how does it cut both strands? If you look at the bottom strand, it also reads GAATTC. So many of the sites that are recognized by restriction endonucleases are what we call symmetric, meaning they read the same on the top strand as they do on the bottom strand. And the result is a fragment that has what are called overhangs that you can see here. In this case, overhang of an AATT, overhang of an AATT. If I take, for example, another DNA fragment from another location that I have cut with EcoR1, and I allow pairing to happen, I can actually allow the overhangs of another fragment to fit into here, as it were. And I can use DNA ligase to put everything together. And so I have made, at that point, a recombinant DNA. And a recombinant DNA is simply a DNA that has pieces that came from from different places. Okay? So instead of bringing it back on itself, that would not be a recombinant, but if I inserted a new piece into there, I would actually make a recombinant. And so that's how recombinant DNA technology is rooted. So restriction endonucleases and DNA ligase. Now, restriction endonucleases themselves are interesting enzymes because in bacteria, they are essentially a protection against viral infection. They're protection against viral infection. So I'll tell you how they work. So if we think about a virus that infects any cell, what it does is it attaches to the outside of a cell, and it injects its nucleic acid into the cell. We've talked about a few viruses so far. When that happens, the nucleic acid of the virus can go into the cell, it can be transcribed, it can be translated to make viral proteins. And so those become the materials to replicate the virus, to package the virus, and then to ultimately exit the cell and go out and make and infect new cells. So that viral DNA has sequences in it that can be recognized in most cases by restriction endonucleases. The restriction endonuclease, for example, GAA-TTC, occurs randomly on average about once every 4,000 nucleotides or so. So if you have a virus that's 40,000 nucleotides in size, there's a pretty good chance it's got one or more of these sequences in it. Some restriction endonucleases recognize sites that are as short as 4 base pairs. Okay? And when that happens, those occur on the order of about once every 256 base pairs. So they can actually chop up a DNA considerably. So if the virus infects a cell that has within it restriction endonucleases, then what happens is the restriction endonucleases are protection because they cut the viral DNA. And when the viral DNA has been cut, it can't be transcribed and translated in an intact form, and consequently the virus is inhibited. Now, restriction endonucleases are found only in bacterial cells. We don't find them in our cells. We don't find them in other cells or other organisms. They're found only in bacterial cells. Now, one of the questions you might have is, well, the bacterial chromosome has 6 million base pairs in the case of E. coli. Surely this sequence must occur in E. coli. And the answer is yes, it occurs throughout E. coli. So then the question becomes, how does E. coli keep from cutting its own DNA? And the answer to that is actually rooted in the nature of the restriction enzyme system. Restriction endonucleases in a cell are always paired with another enzyme. They're called methylases. Methylases. And what these methylases do is they also recognize the same sequence that the restriction enzyme uses, but instead of cutting it, they put a methyl group on it. So in the case of EcoR1, I think the methyl group actually goes on the A. And when that methyl group goes on the A, the restriction enzyme cannot cut it. It cannot cut it. So as you might imagine, the methylase runs around when the cell DNA is being replicated and is methylating that DNA as rapidly as it can so as to prevent the cellular DNA from being cut by the restriction enzyme. The question then arises is, well, what if the methylase gets to the viral DNA before the restriction enzyme does? What do you suppose happens? Well, the viral DNA gets protected. So it's not a perfect system. Everybody wants a perfect system, and it's not a perfect system. But it's a system that works very well and actually does provide quite a bit of protection to bacterial cells against invading viruses. I should also give you a term here, and that term is that a virus that infects bacteria has a name. It's called a bacteriophage. That's B-A-C-T-E-R-I-O-P-H-A-G-E. And some people just simply call them phage. Okay? But that's a virus that infects a bacterium. Well, there are many different restriction endonucleases, and they're actually used considerably as, and here's the methylation process going on here. So there's the target sequence. If this is occurring in a cell, this gets methylated. and it's protected, whereas if it doesn't get methylated, it gets broken up. So if the restriction enzyme got to the cellular DNA before the methylase did, then of course the cellular DNA is going to get cut as well. So there are obviously some potential problems for a cell. Yes, Amy? Yes.
Does that help with mutations? Does it aid in?
Has really nothing. The question is, does this modification have anything to do with mutations? And the answer is no, it doesn't really have anything to do with mutations. So mutations will still be a factor for the polymerase. The methyl group, if that's what your question is, doesn't affect the polymerase in any significant way. But that's a good question, yeah. Okay. Okay. So here are a few very common restriction enzymes. There are hundreds of these that are known. And so if I've got to cut a DNA, I typically I want to cut it into a certain size of fragment. And of course, not every site in a DNA that I want to cut is available at the place where I want it. So I may have to use a different enzyme to get as close as I can to a particular site in order to cut it. So these are commercially available. As I say, there are hundreds of them, hundreds of different specificities, and have really enabled the development of biotechnology, because biotechnology relies very carefully on this. That brings me to the topic here that people, or at least the molecular biologists, clumsily call cloning. Okay? So when I talk about cloning, I want to sort of define that word, because cloning has a lot of meaning that are bandied about. Cloning, as people tend to think of it today, relates to cloning of an organism. So if I decide to clone my favorite dog, I have identical copies of that dog. And yes, in veterinary medicine, that's a very popular idea, is to increase the amount of cloning of pet organisms by people. Cloning, however, technically means identical copies of something. So it doesn't have to be an organism. It can be copies of a cell. So microbiologists know, for example, that when you streak colonies across a plate, what you're doing is you're making individual cells available on that plate that then make a mound of cells, and that's a clone of those cells. They're all identical copies of the first cell that was there. Molecular biologists also use that term to refer to DNA. So multiple identical copies of DNA are also referred to as clones. So unfortunately, that term has been, as I say, bandied about quite a bit. So here I'm going to use the term to refer to making of recombinant DNA, which people call cloning. Okay? They call it cloning because they can make millions and billions of copies of an identical DNA using bacterial cells. So I want to tell you a little bit about how one would go about making recombinant DNAs. How many in you here have made recombinants? Just a few. Okay. So making recombinants in cells has been something that's been practical to do since the late 1970s, with the discovery of restriction enzymes and the purification of DNA ligase, because DNA ligase is necessary to join the ends together to make these things be recombinant. Making of recombinants typically involves using a molecule called a plasmid. And a plasmid, to give you a definition, is a circular DNA that is replicating in a bacterial cell. So that means it has an origin of replication. It is circular, meaning it has no ends. Okay, it doesn't have to be physically a circle, but it's circular. It has no ends. Okay, and it's typically fairly small. So fairly small, a plasmid that we use in the laboratory very commonly has about 2,500 base pairs in it. Some of the larger plasmids might have upwards of 10,000 base pairs in them. Okay, and that's in quite a bit smaller than contrast to, say, a bacterial chromosome, which might have 6 million base pairs in them. The advantage of small ones is that they're fairly easy to manipulate, both in what I'm showing you here and also in being able to put them into cells, because the process of making recombinant DNAs and clones of recombinant DNAs requires our ability to take them out of cells and then to put them back into target cells. And so that's a very important thing. So the smaller that guy is, usually the easier that process is. Well, how do I make a recombinant? I start with something called a plasmid vector. And a plasmid vector always has at least a couple of things. One is that it always has at least a replication origin, meaning that it can replicate in the target cell. The second thing that it typically has is what we call a selectable marker. So a selectable marker is something like a gene that gives either a color or a resistance to an antibiotic, and they're both used. Okay? The most commonly used ones are resistance to an antibiotic. Now the beauty of this is due to the fact that when we go to put these guys back into cells, okay, when I go to make my molecule and I put it back into cells, I've made this guy, and I'll tell you how to do that in a second. Right? I want to put this back into cells so that I can have, for example, the cell make zillions of copies of this DNA for me. Okay? That process of putting it back into cells is very inefficient. Very inefficient. I'm lucky if I get 1 in 10,000 cells, or even 100,000 cells, taking up a DNA that I give it. You say, well, so what? Well, the so what is I've got 99,999 cells I don't want, and 1 cell that has what I do want. If I don't have a way of selecting that, then I play the lottery in whether I get it or not, right? But if I have a gene in here that gives resistance to an antibiotic, then any cells that get this plasmid, or this recombinant DNA, will have resistance to that antibiotic. So if I take my cells, I put the DNAs in, and I then put them onto a plate that has that antibiotic, the only ones that grow will be the ones that got this plasmid. Okay? So it's a really powerful tool to pull that needle out of the haystack. Okay? It's like taking a magnet, and I've got a needle that's magnetic. I just take the magnet, and I pull the needle out of the haystack without having to look through all the haystack to find the stuff. So that's an important thing. Now, there are also things called expression vectors that are related to plasmids that I'll talk about in a bit, and they have at least one other feature I'll talk about too. But before I do that, I want to talk about how we make this recombinant. I can use a plasmid to make a recombinant. I can use an expression vector to make a recombinant. I can use a virus to make a recombinant. All that it takes for me to make a recombinant is a DNA that I can cut and then I can modify by putting something else into. Okay? So in this case, I take the plasmid, I cut it with a restriction endonuclease, like perhaps EcoR1 on the last slide that I showed you, and I take a foreign DNA that has been cut with the same enzyme. So if I have, let's say, a gene for human growth hormone that I have cut with EcoR1, right? That gene might be contained on this fragment right here. I could then mix these guys together and put DNA ligase together and make at least some of the molecules that look like this. Now I say some. Why not all? Well, there's nothing to guarantee it goes in. A lot of them just come right back together and end up looking like this. Everybody understand? This is much easier for it to come back on itself than it is to actually put something in. So now I've got another efficiency to think about. I talked about the efficiency of putting this guy in, but if I put this guy in and I put this guy in, this guy's going to have antibiotic resistance too, right? So some of the cells that I get are going to look like this with a plasmid that looks like this, and some of the cells are going to have a plasmid that looks like this. So I'd like to be able to distinguish this one from this one so that I have another way of pulling the needle out of the haystack. Now, before I tell you about that, I want to make sure that you understand the issues and the problems that I'm talking about here. Do you understand what's going on? Is it Friday afternoon and it's a dark and gloomy day and nobody wants to think about this, or what? Yes? That's actually a very good question. We can do that in some cases, but commonly we only want to have the gene on here because what we're trying to do, it's a very good question, is we want to have a promoter, let's say right here, so that we can transcribe that gene and have the bacterium translate that gene. Okay? It's a very good question. But yes, you can do that, and if you did that, then you would ensure that everything that you had come up had the resistance gene here. You can use this in another way. Let's say I have the plasmid containing a resistance to ampicillin. It's a relative of penicillin. Okay? And I had this fragment carrying resistance to tetracycline. If I grew cells on tetracycline and ampicillin, I would automatically know I had what I wanted, right? Make sense? Okay, so there's some tricks we can use. Now, let's, before I talk about the method, I very briefly want to mention what an expression vector is, okay? So an expression vector is simply something, and there's a variety of things that are in here, but it's simply set up so that anything I put into, in this case I would put it right in this site right here, anything I put into there will be transcribed. So it will have a promoter in addition to the other things that I talked about. It might have a Shine-Dogarno sequence that would help it to be translated. Or I could engineer that on the insert that I'm putting in. Yes, sir?
Just to go back to earlier, what would happen if you tried to introduce that foreign DNA
If you did what?
introduce the foreign DNA
If miscleavage were to occur?
Yeah.
Meaning, okay, so his question is, what if the restriction enzyme didn't cut properly? Yeah, for the most part, you would never see those because that would occur at a very low frequency. And that isn't to say that you don't see odd things in recombinant DNA work sometime, which may involve rearrangements, or it may involve things that didn't quite cut properly. But they're pretty rare, so they're usually not much of a factor. Okay? All right, so that's what an expression vector. So an expression vector helps me to express or to make that protein. So if I've got the human growth hormone and I pop it in this guy right here and I put this in bacteria, the beauty is I can have bacteria making human growth hormone for me. Okay? Bacteria are cheap and easy to grow. And human growth hormone is extraordinarily expensive if I have to use humans to make it, which is the way we used to mess with human growth hormone. Yes, ma'am? So vitamin B12 is not a protein. So vitamin B12 is, so you can engineer bacteria to synthesize vitamin B12. That's correct. But it's not being made by recombinant DNA technology here. Yeah. Okay, other questions? All right, so let's look to figure out how it is that not only can we identify which bacteria get the plasmid, but also which bacteria get the plasmid that I want them to have. Okay? Well, this involves, let me back up here. Sorry. Okay, well, let me just go through this one more time just to show you in a figure what I told you in words. Okay, so here's my plasmid that's got a gene for antibiotic resistance. I've cut it open. I've put the foreign gene inside of there. Okay, and I have inserted into a bacterial cell. That's a very inefficient process, as I said. We use some tricks to get the bacterium to become permeabilized and literally to take it up. And the bacterium will, under some conditions, take this guy up. Once it gets it inside, then we're in pretty good shape because now we've got the antibiotic resistance gene on there. We have a way of flagging this. So if I grow it in antibiotic in medium that contains the antibiotic, then I get this guy, and all the other guys die. Okay, and so that, of course, I get zillions of these after because I double every time. So I can get a lot of bacteria that have the thing that I want by growing them literally overnight. Okay, now let's talk then about how it is that I get the thing that I'm after. Now this involves use of a gene called the Beta-Galactosidase gene. You've heard of that gene once before when I talked about the lac operon. So the Beta-Galactosidase gene is one of the genes of the lac operon, and I need to tell you what it does. So the Beta-Galactosidase gene cuts the molecule known as lactose. Lactose is a sugar. Lactose contains galactose and glucose. So it contains two sugars, galactose and glucose. When bacteria encounter lactose, as I said, they want to use that. And so the way they use it is they synthesize the operon, and the operon makes this gene right here so that it can cut lactose into two pieces, galactose and glucose, and the bacterium can use that. It can't use the lactose unless it cuts it up with this enzyme. Okay? So this enzyme is very good at that. So I have my expression vector that is set up with several things. It's, of course, got the ampicillin resistance, it's got a replication origin, and it's got a gene for beta-galactosidase production. Okay? Now, biochemists, I like to say, are lazy people. And they're lazy people because we like to do things the easiest way that we can. And there's some very good reasons for that. We're human beings. Human beings like to do things the easiest way that we can. And so most biochemists are human beings. So, okay, guys, all right. Anyway, so what is beta-galactosidase? It cuts lactose. Well, biochemists being lazy say, well, it'd be nice if I could fool the enzyme. Remember how we fooled chymotrypsin to get the color change? We produced a color when chymotrypsin acted, right? We saw that yellow color being produced. Well, it turns out that beta-galactosidase can also be fooled to cut a molecule. The molecule that beta-galactosidase gets fooled by is one that you should know. It's called X-gal, the letter X-GAL. Okay? X-gal is a molecule that, X-gal is a molecule that is blue. And when it gets cut by the enzyme, it loses its color. You can think of it as becoming white. So we have blue versus white. Okay? If I take this plasmid and I have the coding for the beta-galactosidase gene, let's say I just took this plasmid and I put it into bacteria, and I gave the bacteria X-gal, an antibiotic. What would I see? Well, I would see, first of all, that only the bacteria that got the plasmid would grow, because the ampicillin would ensure that. And when I gave the bacteria X-gal, they would produce a blue color. Right? Because, I'm saying it wrong. They would produce a white color because the blue that it would start with would get cut, and I would have white as a result of that, right? So the X-gal is going to get cut if the enzyme is present. If I were to take a plasmid that didn't contain this, and I put it into bacteria, and by the way, I'm using bacteria that don't have the lac operon. So the only way they have this gene is if we put it in with a plasmid. The bacteria don't have the operon at all. We've taken it out of them. Okay? If I give them a plasmid that doesn't have that gene, and I do the same experiment, what am I going to see? Well, the ones that have the plasmid are going to grow, but because this gene isn't present, when I give them X-gal, they're going to turn blue. Everybody follow? So blue means they've got no enzyme. White means, blue means they've got no beta-galactosidase. White means, why can't I say that right? Blue means that they have no beta-galactosidase. White means they have no beta-galactosidase. Right? I said it backwards again, didn't I? You know, I do this every time. I have a lecture on YouTube. It's always embarrassing when you get in these things, because these are all going on YouTube. They're seen by people all over the world. Hello, world, right? And here's the professor, and he gets up there, and he goes, oh, no, I said it backwards again. So I've done it one more time. But at least this time I caught myself in the middle of saying it. If they have the enzyme, we're going to see white color, because we're going to break the X-gal down. If they don't have the enzyme, we're going to see blue color, because they won't be able to break it down. Yes? Well, I'm saying I had two different plasmids. One plasmid that had the beta-galactosidase, that will give me white color. One plasmid that doesn't have the beta-galactosidase, that's going to give me blue color, because the bacteria have already taken the enzyme away. It would be, whether it's considered foreign DNA or not, if I give a plasmid to a bacterium that didn't have a plasmid in the first place, that is foreign DNA by definition. Okay? Okay. Now with that confusing intro, let's go on. All right. Well, what we want to do is we want to put this guy into a plasmid, and we want to put that plasmid into the cell, and we want to know which cells have this inserted DNA and which don't. So what do? Well, we insert this DNA into the middle of the beta-galactosidase gene. When we do that, we're going to destroy the gene. We're going to destroy the gene when we put it in the middle of the gene. So those that get this thing are going to lose beta-galactosidase. What color are they going to be? Blue? Did I just get it backwards again? Inserts have white. Inserts have white. Every time I do this lecture, I do the same thing. So, damn. Damn, damn, damn. Let's just come back to it. No, we're not just going to come back to it. We're not going to do that. All right. Color. And then cells do not produce blue color, but are resistant. Okay. So I said it backwards. Okay. The problem is that X-gal, when it gets cut by beta-galactosidase, is blue. That's why I couldn't. Why I've got it all backwards. Okay. So bacterial cells, when they the X-gal, when it's cut by beta-galactosidase, produces a blue color. Okay. Produces a blue color. If there's no beta-galactosidase, you will have a white color. Okay? All right. If I'd started with that, I would have been in good shape. All right? So X-gal, all right, produces a blue color when beta-galactosidase is present. So let's go back and start our thing up here. So if I have beta-galactosidase present, that I have this plasmid shown here, I put it into bacteria, I'm going to get a blue color. If I take this out of here and I put it into bacteria where I have no beta-galactosidase, I'm going to have a white color. Okay? Are we now clear? This means I can't ask you this now in a test, right? Too much confusion over the thing. All right. So how does this work if we put a plasmid in here? We cut the beta-galactosidase in the middle. We insert our gene. We destroy the beta-galactosidase. When we destroy the beta-galactosidase, we have the situation that we had over here. No gene. But remember that we have a mixture. Some of these guys are going to have insert, and some of these guys are not going to have insert. If this guy, we cut it and comes back on itself, we make this, right? If this guy, we cut it and we insert and we get this, then we got two different things. So we have a mixture of these plasmids and these plasmids. That means some of them have the beta-galactosidase functional. That'll be the ones in the top that have no insert. And some of them will have beta-galactosidase non-functional, all right, which will be those that do have an insert. All right? Take our mixture, put them into E. coli cells. We get three possible things. Okay? One possibility is we have those that get no plasmid at all. Vast number of cells get no plasmid at all. They're going to die when we put them on antibiotic. We have those that have no insert, which means they have a functional beta-galactosidase. They're going to produce a blue color. And those that have an insert are going to be antibiotic resistant, just like these guys are. However, they're going to have a white color. So we look for colonies that are white, pick white colonies, okay, and analyze those because those are most likely to be the ones that have the insert that we want. The first one is cells that get no plasmid at all. So they don't have any antibiotic resistance. They die. We never see them. In fact, you can see this is an empty plate down here. Those that get a plasmid but have a plasmid that has no insert will be blue, and those that have a plasmid that have an insert will be white. Now, in reality, we don't have three different plates. We have them all on one plate. Some of the colonies will be blue, some of the colonies will be white, and of course, the dead ones we never see anyway. Right? All right. Sorry for the confusion. Yes? Yes. So these are both antibiotic resistant. That's the only way we get growth. It's kind of hard to see on there, but those are white colonies that are on there. Okay, so the blue ones show up a little bit more easily on this thing. So this is called blue-white screening, right? So blue-white screening allows us to see if we have the insert or not. Now, I told you I took the lac operon out of the cells. You might remember that we had something that acted to enhance the transcription of the operon, right? What acted to enhance? Okay. Well, one of the things that we needed was we had to have the, excuse me, we had to get rid of the lac repressor. Okay? We had to have an inducer molecule, right? Well, if we use lactose as an inducer molecule, excuse me, and we are breaking down lactose with this enzyme, we're going to be destroying our inducer molecule over time, right? So over time, we're going to see less and less of what we're after. So we may have variable results with that. So because of that, scientists have made another artificial molecule. In this case, is an artificial inducer. It's a molecule called IPTG. The beauty of IPTG is it will take the lac repressor out of the picture, okay? But it won't be broken down by the beta-galactosidase. Consequently, it's a very strong inducer of the operon, and with this, you can make a ton of whatever happens to be in here. That's a very powerful tool for us to use. Okay? Okay. Sorry. All right. What else can I show you here? Here's my favorite, okay? This guy right here, okay? How many of you have ever seen an array before? Okay. So what is an array? Well, let's imagine, if you will, that I have the genomic sequence of an organism. And we have the sequence, the complete sequence of the genome of thousands of organisms now, bacterial all the way up to humans. Let's say that we have the sequence of human DNA. And we also know within that organism the sequence of every single gene in the human genome. The sequence of every single gene. Okay? There's 20,000. What if I said, is it possible for me to go to a laboratory and have somebody synthesize, using chemicals, the sequence of a gene? The answer is, it turns out to be fairly easy to do. We can do this fairly readily, or at least pieces of a gene. Okay? So I can go and say, here's a sequence of a human gene. Can you make me this in the laboratory? And the person says, sure. How many do you want? Oh, I don't know, a few million copies, which sounds like a lot, but it's not that much. Okay? And I take that sequence that they've made me in the laboratory, and a few million would fit in a spot that would be smaller than what your eye could see. And I take and I chemically bond it to a specific location on a glass plate. So I have, let's say, the gene for DNA polymerase that I've taken and I've put right there on that spot. And I've got millions of copies of that same thing on that one spot. Everybody with me? Well, let's now imagine I do that for every single gene in the human genome, one spot corresponding to each gene. Okay? That's pretty cool. What I would like to know is how much of any given gene is being made in a human cell at any given time. Well, I made this cleverly, so I made it complementary to the actual messenger RNA that's going to be made. It's complementary to it. So they'll form base pairs. I'm simplifying this to some extent, so bear with me. Okay? Let's say I take and I now have a liver, set of liver cells, and I want to know what genes are being made in liver cells. I take those liver cells, I bust them open, I isolate the messenger RNA, and I dye all the messenger RNA green. I can put a green dye on every one of those. I can put that mixture of all those messenger RNAs with all those green dyes on there on this plate, create conditions for base pairs to find each other, and believe it or not, they will find each other. Okay? The beauty of this is that the amount of green that I see is a function of how much of that messenger RNA was being made, because it's going to base pair with its specific complement on there. And if I know that this is DNA polymerase, and I know this one is a ribosomal protein, and I know this one is beta-galactosidase or something like that, I can then go back and say, well, in this liver cell, the following genes are being made. They're being made, in this case, in high quantities because I can see a bright spot, or they're being made in very low quantities because I can barely see the spot right here. Are you asking about the colors? Yeah, I'm getting to that. Okay, I knew that was the first thing that pops up. Well, what about the other colors? Why do you see red? Why do you see yellow? Let's imagine that I not only had a liver cell, or liver that I was interested in, but I had a person who had liver cancer. And that person who had liver cancer, I isolated the tumor from that person with liver cancer, and I isolated the messenger RNA from that, and instead of having it be marked green, I marked it red. What would happen? Well, what I would see, I did the same experiment, but now I mix the messenger RNAs from the green and the red, and I put them on this plate, and I let them find their complementary region. What would I expect to see? Well, I would expect to see that some of the genes would be made in both cells. Let's say DNA polymerase. Both cells need to have DNA polymerase being made, and probably in about the same quantities. But I might expect that some genes would be made in the cancer cell and not in a normal cell. Other genes might be made in a normal cell, not in a cancer cell. And a whole bunch of genes that might have varying amounts, higher in one or lower, etc. Well, what I can do then is when I mix this, by the way, when you do these experiments, red plus green equals yellow. So where you see yellow, you see genes that are being made in equal quantities in each of those cells. When I see something that is redder than it is greener, okay? Whatever, I'm not very good with color here, okay? When it's more red, it means it's being made more in the cancer cell. If it's more green, it's being made more in the liver cell. And not only that, but the intensity of each one tells me how much of each one is being made. Now, the beauty of this is, first of all, I know which genes are preferred in one cell type versus the other. They provide targets for drugs. What if I knock out this gene right here that's being made a heck of a lot in the cancer cell, and I know what it is, and I know the cancer cell needs it. What if I target that gene with my drug? I've got something that is going to hit that cancer cell pretty darn well. Okay, did you have a question, Andrew? There's other things that you can use, yeah. Well, but you're not doing this by eye. The computer's doing all this. So this is actually a relatively simple one. You could have one of these. This doesn't have 20,000 genes on it, okay? But you could have one of these that does have 20,000 genes. The computer would automatically go through. It would tell you for each gene which one it was preferentially being made in. It would also tell you if it was made or not. Maybe both of them don't make it for whatever reason, okay? So no, the computer's going to do this because it's way faster and it's way more accurate than you're going to do by eye. Yeah. Okay? So this is a really powerful tool. Imagine that you say you're a drug company and you're saying, I've got this new drug that is preventing blood clotting. I've got a new warfarin, right? It's a very popular thing to make blood-thinning drugs. I've got a new warfarin. I want to know, is this going to have a detrimental effect on, I don't know, bone cells or liver cells or whatever? What would I do? Well, I would take bone cells. One group I would treat with the drug. The other group I would not treat with the drug. One would get a red color, one would get a green color. I'd do the same experiment, and I'd ask the question, do I see a significant difference in pattern in the drug-treated cells versus the other cells? And I would know not only is there a difference, but which genes are affected. So I could say, is that critical? And isn't that interesting why that's happening? Okay? So there's some really remarkable things that arrays can tell you. So what I'm describing to you, what I've just described to you, that I've described to you right here is something we call transcriptomics. Transcriptomics. It tells us what the transcriptome is. And the transcriptome is the collection of genes being transcribed. The transcriptome is the collection of genes in a cell that are being transcribed. There's all kinds of things. We've talked about proteomics so far. We did a similar experiment with the 2D gel, where we could see whether the actual physical protein was being made or not in one cell versus the other. In this case, we're looking at the messenger RNA. What if we compared the two? Will we expect to see a difference between the two? We might. What if a protein is being made but being broken down, for example? We can see that difference very readily if we compared the proteome of a cell to the transcriptome of a cell. Okay. You guys got it? Questions? Okay, you look like you are tired. I have a reward for you. So I see a lot of empty seats here today, so I think it'd be a good day for extra credit. Doesn't have to be a nice day. And I will tell by whose exams are left over, so you don't even have to do anything. Oh, should we do a song? Or do you want your exams? Song? How many want song? How many want exam? Song? What's that? Why not both? Wait, no, it's not either or. Okay, let's quickly do a song. I've got a cute song that we can do. Okay. So today's song is to the tune of Chim Chim Cheree. It's an old 60s song. It goes. It's about restriction enzymes. I'm obsessed with AAGCTT because it's the binding site of Hindi3, cutting up DNA most readily. The ends are not blunt when they're cut up, you see. 5' overhangs of AGCT. Bacteria don't have an immune system, so they must fight off phages or they will not grow. Protection by copying is their strategy, and one of the cutters we call Hindi3. On binding of AAGCTT, the site recognition sites bent easily. Phosphodiester attacking, meanwhile, has water behaving as nucleophile to stave off the phage for a little while. Why don't these enzymes cut cells' DNAs? The answer's provided by amethylase. Adding a methyl group on top of what? The sequence these enzymes would otherwise cut. So cells get protected in this simple way from nuclease chewing of their DNA. Phage is not lucky in most every case unless methylases win the enzyme race. If that happens, then the cell gets erased. Da, 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 da.